అది బ్రేక్ అవుతా ఉంది. ఓకే, ఎనీవే. Yeah. So I'll share my screen here. ఏ స్క్రీన్ కనిపిస్తా ఉందా?
Yes, sir.
Okay. Yeah. So we were discussing about enzyme inhibition. So enzyme inhibition is nothing but some material which is chemical compound which decreases or stops the biological activity of enzyme, and they disturb the complete sequence of enzyme substrate complex, and they avoid that interaction between enzyme substrate complex and enzyme substrate complex does not occur, and ultimately they prevent the formation of product. So these enzyme inhibitors can be of three types: competitive, non-competitive, and feedback. So competitive, as we discussed earlier, they compete with the substrate. Okay, so this is the inhibitor. So they are similar in shape and size to the substrate as well as the active site. They come and they block, they bond the form on the active site, and they inhibit the enzyme substrate complex, and that is how no product is formed. So this can be, it is reversible. It can be controlled. How we can control means we can increase the substrate concentration and we can avoid the effect of competitive inhibition. That is the first case. Second case is the non-competitive inhibitor, which does not come and combine with, bind with the, or it does not compete with the substrate. So this is the inhibitor, competitive inhibitor, which is not similar to the active site or the substrate shape. But what happens here is this inhibitor, it comes and combines with the enzyme at some other site, which is the allosteric site. So by joining here the non-competitive inhibitor at the allosteric site, the conformational change occurs in the enzyme at the active site. The active site gets changed, and it is not compatible with the substrate, and the enzyme substrate complex does not occur. So that is how product is not formed. And one more thing is that we cannot control this. Even though we increase the substrate concentration, this cannot be controlled. Okay, it is irreversible. Coming down to the third type, that is the feedback inhibition. Feedback inhibition is a type of allosteric or non-competitive, allosteric or non-competitive shell. So, as we discussed earlier, that this feedback inhibition, it can be of either the allosteric or non-competitive type. That is the second type, which occurs in the biochemical pathway in order to control the amount of product metabolic pathway. So what happens here is this what is feedback inhibitor? What does it do? It comes and combines with the product. Okay. There are these feedback inhibitors. They participate in a reaction where multiple enzymes are involved, and then intermediate products are formed. These type of reaction, those metabolic pathway type of reactions, these type of feedback inhibition take place, participate. So they that can be allosteric or non-competitive type. So this occurs when there is a buildup of or excess of product, but the metabolic need declines. Cells use this method to maintain balance or stasis. Sometimes what happens here is excess amount of product is that particular metabolic reaction occurs, and more number of product is produced here. more than what is required, more number of product is excess product is produced. Okay, this occurs in sometimes in cells. This method is known as homeostasis. Homeostasis process. So the final product in a series of reactions, it inhibits an enzyme from an earlier step in the sequence. What happens here is whatever the final product has been produced, suppose there are 10 reactions occurring in a particular metabolic pathway. So after the 10th reaction, the final product is produced. So after a series of 10 reactions, final product is produced that inhibits an enzyme. So that product itself inhibits an enzyme from an earlier step. It can inhibit the enzyme 1, enzyme 2, enzyme 3, anything. Okay. So it acts as a inhibitor in an allosteric or non-competitive pathway, non-competitive inhibitor type fashion, and it combines with that particular enzyme at some allosteric place, and that is how the further reaction is uh, stopped. So the product binds at an allosteric site and temporarily inactivates the enzyme by a non-competitive inhibition. That's what I said. So as the enzyme can no longer function, the reaction sequence is altered and the rate of product formation is decreased. So this diagram will be clear for you. So suppose there are four reactions here. Okay? Reaction one, reaction two, reaction, reaction four. So this is the first point where the substrate binds with the enzyme one, the product is formed, then another enzyme binds with this substrate, this product acts as the substrate and this enzyme combines with this. Second product is formed. Then again third enzyme, this combines with this. Okay. Another product is formed. Then again enzyme 4 combines with this substrate. The end product is formed. Now what happens here is feedback inhibition. So, so in feedback inhibition, the final product inhibits the enzyme 1. So here what happens here, this product itself acts as a feedback inhibition. So this goes and it combines with enzyme 1 at some allosteric point. So ఎక్కడైనా ఉండొచ్చు ఎంజైమ్లో allosteric సైట్ ఎక్కడైనా ఉండొచ్చు. సో యాక్చువల్లీ ఎంజైమ్ 1 ఏంటి, ఇక్కడ ఒక సబ్స్ట్రేట్తో బైండ్ అవుతుంది ఈ పర్టికులర్ పాయింట్. దిస్ ఇస్ ది యాక్టివ్ సైట్. బట్ మనకి ఇక్కడ ఫీడ్బ్యాక్ ఇనిబిటర్ ఎట్లా పనిచేస్తుంది, ఒక ఎంజైమ్లో ఎక్కడో ఒక చోట అంటే నాన్ ఇనిబిటర్ టైప్ అంటే అల్లోస్టెరిక్ ఫ్యాషన్లో ఎక్కడో ఒక చోట ఎంజైమ్కి కంబైన్ అయిన తర్వాత అది ఎంజైమ్ని ఇన్యాక్టివ్ చేస్తుంది. అంటే ఈ యాక్టివ్ సైట్లో కన్ఫర్మేషనల్ చేంజ్ జరిగి ఈ సబ్స్టేట్ అనేది ఇక్కడ బైండ్ కాదు, తద్వారా ఎంజైమ్ అనేది ఇన్యాక్టివ్ అయిపోతుంది. సో నో ఫర్దర్ రియాక్షన్ అంటే ఇక్కడ నెక్స్ట్ ప్రోడక్ట్ అనేది ఏర్పడదు. సో ఇట్లా ఈ రకంగా రియాక్షన్ జరిగినట్లయితే దీన్ని ఫీడ్బ్యాక్ ఇన్హిబిషన్. సో ఇక్కడ ఫీడ్బ్యాక్ ఇన్హిబిషన్ ఏంటంటే ఈ ప్రోడక్ట్ ఎక్కడైనా కంబైన్ కావచ్చు, ఫస్ట్ ఎంజైమ్ దగ్గరే కావచ్చు. ఇట్ కెన్ కంబైన్, ఇట్ కెన్ బైండ్ విత్ ఎంజైమ్ 2 అండ్ పర్ఫార్మ్ ది సేమ్ రియాక్షన్, లేదా ఎంజైమ్ 3 దగ్గర వచ్చి సేమ్ రియాక్షన్, లేదా ఇట్లా ఎక్కడో చోట మల్టిపుల్ ఎంజైమ్స్ ఇన్వాల్వ్ అయిన చోట పాత్వేలో ఈ ప్రోడక్ట్ అనేది కంప్లీట్గా ఫీడ్బ్యాక్ ఇన్హిబిటర్గా పనిచేస్తుంది. It goes back and combines with any intermediate enzyme and at a allosteric point, and that reaction completely stops. No substrate formation, enzyme substrate formation does not occur. Okay, so that is how the further reaction is stopped. So enzymes, going to sum up, enzyme inhibitors prevent the formation of enzyme substrate complex and prevent the formation of the product. Product does not occur. Okay, whenever a inhibitor is involved in. So these inhibitors are of three types: competitive inhibitor, non-competitive inhibitor, feedback inhibitor. So inhibitor, competitive inhibition, inhibitor and substrate here what is happening in the first case is inhibitor and substrate both are similar to the active side. That's why they compete. That's why the inhibitor is a competitive inhibitor, which is competing with the substrate. So with each other to bind at the active site of the enzyme. When inhibitor binds to the active site, it forms stable enzyme inhibitor complex and reduce enzyme activity. Whereas second case that is in non-competitive inhibition, inhibitor molecule binds at a site other than the active site. So that site is known as allosteric site. So the binding of the inhibitor to allosteric site causes a conformational change to the enzyme's active site, and that is why the enzyme activity is stopped. So obviously, when the conformational change occurs, it does not substrate cannot come and bind here. That is how the enzyme activity is stopped or it becomes inactive. So you have to remember two things. One thing is that in the second case, that is a non-competitive inhibitor, the inhibitor is binding to the enzyme at some other particular site, which is known as allosteric site. Whereas second point is that it is not similar to the substrate molecule. So that is why it is we are calling it as non-competitive inhibitor. Okay. So that is second case. Third case is the feedback inhibition. Feedback inhibitor. Okay. So that inhibitor is acting on some previous enzyme. You can take it that. So feedback inhibitor is a type of allosteric inhibition occurring in metabolic pathways of living organisms. That may be any metabolic path. So the where multiple enzymes are involved in multiple products are formed, intermediate products. So the final product, if in excess, is a series of in a series of reactions. Maybe it's an enzyme from earlier step, the sequence, and decrease the speed of the reaction. So how this feedback inhibition is acting? So in a pathway where so many multiple steps are involved, step one, step two, step three, step four, and so on. But the end product which is formed here, this end product, it goes back and it combines with some previous enzyme at a allosteric point, and it becomes the conformation change occurs at the active site, and the substrate won't bind with this enzyme, and the enzyme becomes inactive. And the further reactions are blocked here itself. Okay, this may occur at any stage of a multiple reaction. It may occur at the stage 1, stage 2, or stage 3, and so on. So, product itself acts as a inhibitor. We call this as feedback. Okay, so this is all about feedback enzyme inhibitors. So, which is very important for short answer, or you can say essay question also can be asked for this. So, we have to explain all the three: competitive inhibitor, non-competitive inhibitor, and the feedback. Okay, so keep this thing in mind. Then now we will go to the topic that is the factors controlling the enzyme action. So this is the last topic in this enzyme chapter, that is the factors affecting the enzyme activity. So here if you see, so this is the enzyme catalyzed reaction where this is the substrate, this is the product. So in any reaction, if you see there are two reactions, as we have discussed this figure, the graph earlier also. So to know the importance of activation. So here for any substrate getting converted to product, it has to reach it utilizes a lot of energy that is known as the activation energy, which that is the point where the substrate starts
Sir,
getting
sound in plus.
yes, just a
Sound
minute.
in plus.
Yes, yes, ఒక్క నిమిషం. Hello. Yes, now is it audible? వినిపిస్తా ఉందమ్మా? నా వాయిస్ వినిపిస్ ఎస్ నా వాయిస్ వినిపిస్తా ఉంది అందరికి. ఎస్ వినిపిస్తా ఉందా? ఎస్ ఆర్ నో చెప్పండి.
Yes, sir. Yes, sir. Yes,
ఓకే. ఓకే, నో ప్రాబ్లం. ఐల్ కంటిన్యూ. ఎక్కడైనా మీకు ప్రాబ్లం ఉంటే చెప్పండి, నేను ఐల్ చెక్ ఇట్. సో ఇక్కడ ఈ గ్రాఫ్ లో ఒకటి గుర్తు పెట్టుకోండి, ఇన్ ఎనీ టైప్ ఆఫ్ రియాక్షన్ ఒక సబ్స్ట్రేట్ అనేది ప్రోడక్ట్ గా ఏర్పడాలంటే ఒక పర్టిక్యులర్ అమౌంట్ ఆఫ్ ఎనర్జీ అనేది అవసరం ఉంది. సో ఆ పర్టిక్యులర్ అమౌంట్ ఆఫ్ ఎనర్జీని మనం యాక్టివేషన్ ఎనర్జీ అంటాం. సో ఇక్కడ ఈ రెడ్ కలర్ గ్రాఫ్ మీరు చూసినట్టయితే దిస్ ఈస్ టేకింగ్ ప్లేస్. సో ఇది ఇక్కడ వరకు పోతా ఉంది. సో దిస్ ఈస్ ది పాయింట్. సో ఇక్కడ ఏంటి, దెర్ ఈస్ నో క్యాటలిస్ట్ ఇన్వాల్వ్డ్, అంటే దెర్ ఈస్ నో దిస్ రియాక్షన్ ఈస్ టేకింగ్ ప్లేస్ వితౌట్ అన్ ఎంజైమ్. ఒక ఎంజైమ్ లేనట్లయితే సబ్స్ట్రేట్ ఇంత హ్యూజ్ అమౌంట్ ఆఫ్ ఎనర్జీ ఇక్కడ ఇంతవరకు ఇంత హ్యూజ్ అమౌంట్ ఆఫ్ యాక్టివేషన్ ఎనర్జీ యూజ్ చేసుకుంటేనే అది ప్రోడక్ట్ ని ఏర్పరుస్తుంది. సో ఈ పాయింట్ ని ఏమంటామంటే ట్రాన్సిషన్ స్టేట్ అంట. ట్రాన్సిషన్ స్టేట్ అంటే ది సబ్స్ట్రేట్ స్టార్ట్స్ గెటింగ్ కన్వర్టెడ్, చేంజింగ్ ఇట్ సెల్ఫ్ ఇంటూ ప్రోడక్ట్. సో దిస్ మచ్ అమౌంట్ ఆఫ్ ఎనర్జీ ఈజ్ used. But if you see the second reaction, where this blue color reaction graph, where only this much amount of energy is used, this is taking place with a enzyme. So substrate in presence of enzyme, what is happening here? Enzyme lowers the activation energy drastically, and the reaction takes place at a very high pace, and more products are formed in a very short time. So that is the difference. So enzyme catalysts reduce the activation energy required, thereby increasing the rate of reaction. So the basic enzyme catalyzed reaction can be represented as, so enzyme catalyzed reaction is evident. So this is the substrate, presence of enzyme, enzyme substrate complex is being formed, and the after the reaction enzyme substrate complex, we are getting the end product plus the enzyme. The enzyme is freely released without being altered either quantity wise or quality wise. So it just catalyzes the reaction by lowering the activation energy of that reaction, whereby it speeds the rate of reaction at a very high pace. So this is a forward reaction. Same thing is applicable for reverse reaction. So, so it is enzymes favor reversible reactions. So they favor forward as well as backward reaction. Same equation, you can take it this way. Suppose this product is the substrate, combines with the enzyme substrate complex is formed. Again, the substrate that is here, that acts as the product, and the enzyme is released. So keep this thing in mind. Another thing is that substrate binds to the active site of the enzyme, forms enzyme substrate complex, and finally release the product to yield back the enzyme. That is, the enzyme is freely released. And enzymes accelerate reaction by how lowering the activation energy. So more and more substrates can reach transition state and form products. So factors affecting the enzyme action. Just a minute, somebody is voting. So factors affecting the enzyme activity here, a number of factors affect the enzyme activity of an enzyme in an enzyme catalyzed reaction by majorly affecting the tertiary structure of protein, that is the active site. Okay. So there are number of factors that are involved. The enzyme activity. Enzyme activity అంటే ఏంటి? Catalysis. ఏం చేస్తాయి? They catalyze the metabolic reaction or biological reaction. So how they affect is that they affect the tertiary protein structure. So protein structure ఏదైతే 3D structure which is formed because of the long amino acid chains. So so that particular tertiary structure is the active site where the actual substrate comes and binds. So what are those factors? The main factors are temperature, then the hydrogen ion concentration, that is the pH, then the substrate concentration also affects, and also the presence of inhibitors also. So these are the few main factors which affect the enzyme activity. So if you see the effect of temperature on enzyme, first one. So enzymes are thermolabile. They are sensitive to temperature variation. So as we discussed in the properties that enzymes they need an optimum temperature. So each enzyme is most active at a specific temperature called optimum temperature. Example is 37 degrees for amylase. Okay. So normally the range of optimum temperature for enzymes is generally is between 25 to 40 degrees centigrade. So optimum temperature means the suitable temperature where the enzyme acts at its full potency. So 100% active గా పనిచేస్తుంది. So that range is known as the optimum temperature. But uh, if at all any change is there, so this optimum temperature that can be different for different enzymes. So for example, here 37 degrees mileage enzyme. It acts very actively at this temperature. So this is the optimum temperature for amylase. Like this, there is that, but any particular enzyme has its own optimum temperature. So that optimum temperature range is between 25 to 40 degrees centigrade. So whenever the temperature increases up to optimum temperature, increases enzyme activity due to increase in kinetic energy that molecules possess. Thus, rate of reaction also. So what happens here is whenever there is an increase in temperature more than the suppose optimum up to optimum temperature. So slowly when we increase the temperature up to optimum temperature, what change occurs in the enzyme? The kinetic energy it increases the enzyme, the molecules. So kinetic energy, so the enzyme activity due to increase in kinetic energy that molecules possess. So molecule, the kinetic energy in the, is increased in the enzyme, and that is how the enzyme activity is increased. Then up to what point up to an optimum temperature of that particular enzyme. And this slowly the rate of reaction starts increasing. So it is directly proportional. So increase in rate of reaction is directly proportional to increase in temperature up to optimum temperature. So increase in temperature beyond the so ఇక్కడ beyond the optimum temperature range or a higher temperature puts a strain on the bonds, the tertiary structure of protein like the hydrogen bonds, then the disulfide bonds, and so etc., leading to the loss of tertiary structure, thereby changing the shape of active site and ultimately loss of enzyme activity occurs. This is known as denaturation of enzyme. What happens here is whenever the temperature is increased beyond the optimum temperature, the it starts its effect on the bonding. Okay, whatever hydrogen bond or disulfide bonding is present in the tertiary structure of protein, which is made up of long chains of amino acids. So those bonds start breaking down because of the increase of temperature. And slowly, the shape, that is the tertiary 3D structure of that protein or that enzyme starts changing, and the active site that are present, that completely changes, thereby uh, making the enzyme inactive. This action is known as the denaturation enzyme, or you can say protein coagulation occurs. So as the at high temperature, protein coagulation occurs, and denaturation of enzyme is formed. Why protein means enzymes, that 3D structure is made up of protein only. The effect of temperature on enzyme, generally, if you see here, generally the enzyme activity increases from 5 degrees to optimum temperature and double with every 10 degrees rise in temperature. So this rise in activity, enzyme activity, it starts from 5 degrees. So 5 degrees, which you see here, 5 degrees, the enzyme activity starts increasing up to what? Up to optimum temperature. And then this increasing is occurring at what pace? It is getting doubled with every 10 degrees rise in temperature and decrease beyond the optimum temperature. So this is called temperature coefficient, Q10 for most of the biological processes is double. So here, if you see here, this is the rate of reaction, x-axis and y-axis, the temperature. So here, what is happening here? Gradually, we are increasing the so heat energy causes more collisions between enzyme and substrate. So the rate of reaction is increasing. So as you increase the temperature, the rate of reaction is also increasing up to what? Up to this is the optimum temperature for humans is 37 degrees. So after this point, the enzyme denature at temperatures at high temperatures, so rate falls rapidly. So after this point, that is 37 degrees centigrade, whenever you increase further, so when you start increasing further temperature, what happens here? The rate of reaction starts falling down. So why it is falling down or the enzyme activities decrease? Because after the optimum temperature, that is 37 degrees, the enzyme 3D structure is changed. So because of the denaturation, protein coagulation, denaturation of enzyme becomes inactive and slowly the rate of reaction decreases, even though you increase the temperature beyond the optimum temperature. So that is how enzyme acts on the enzyme. Sorry, temperature acts on the enzyme. Now coming down to second one, that is the effect of pH. So pH is the hydrogen ion concentration. So pH depends on the concentration of hydrogen in the solution. So each enzyme has its own optimum pH range from 6 to 7.5. So as you know, 7 is the neutral, and below 7, that is 5, 4, 3, 1 are acidic.
Sir, voice is not coming, sir.
Yes, I'll check once again. Yes, will this sound good?
No, sir, I can't hear, sir, voice.
Yes.
Is it clear now? Yes, voice will be sound. Anybody respond?
No, sir.
Voice is coming. You check your signal. Tarak,
వినిపిస్తుంది సార్.
voice is clear. You just check your signal. Signal ఉన్న చోట కూర్చో. Others respond. Voice వినిపిస్తా ఉందా?
వినిపిస్తుంది సార్.
Okay. Yeah. So I'll continue. So enzyme has an optimum pH range which is between 6 to 7.5. So as you all know, 7 is the neutral point, and below 7 it is acidic. That is 5743. Sorry, 54321, and beyond 7 is basic. Basicity, that is 8, 7, 8, 9, 10. So enzymes are denatured at extremes of pH. Okay, whenever there is fluctuation between the optimum pH range, then the enzymes start getting denatured. Except for a few enzymes like pepsin, which can function at pH levels as low as 2, which is acidic pH.
Sir, no, sir. I can't hear anything, sir.
Yes, you check your or you just log out and just log in once again, because others can hear clearly, and even I can check it that voice is audible. So do one thing, you just log out and log in once again, or you check your signal strength and sit where there is a signal is strong. Okay. Yeah. So another enzyme is trypsin, which works at a pH of around 8.8, which is highly basic. Okay. So except these few enzymes like the pepsin, which is active at low pH of 2, and the other enzyme, that is trypsin, which works at a high pH of around 8.8. But most of the enzymes operate maximum at neutral pH, like amylase, which acts at around 6.8, then the urease, which acts around 7, catalase, which also acts at the 7. So normally the neutral range means what? 7. So most of the enzymes, they catalyze, or their activity is more at this range of 7, like the amylase 6.8, urease, catalase, and 7. Then the change in hydrogen ion concentration causes change in the ionization and solubility of enzymes. And pH also affects the formation of hydrogen bonds and disulfide bonds. So what happens here is, whenever there is any change in hydrogen ion concentration beyond the optimum range, so that has a direct effect on the ionization part as well as the solubility of the enzymes. So that is how the pH affects the formation of, how it affects means it forms the direct effect you can see in the formation of hydrogen bonds and the disulfide bonds. So here you can see here, this is the graph which represents the pH, and this is the enzyme activity. So normally the optimum range is around 7. Okay, not 8, it is 7. Neutral, this point is the active. So there are few enzymes where optimum range may be low, below 7, and there are few enzymes which are active at beyond 7, which is, this is basic and this is acidic. Then the third factor is the effect of substrate concentration. So at constant enzyme concentration, increase in substrate concentration increase the rate of reactions. So whenever the enzyme is constant, so enzyme concentration is fixed, and we increase the substrate concentration, thereby the rate of reaction is also increased. That means the rate of reaction is directly proportional to the substrate concentration. So as we increase the substrate concentration, rate of reaction also increases at constant enzyme concentration. So the reason is because more and more active sites of the enzyme will be occupied by substrate molecules. This will happen till all active sites are occupied by the substrate known as saturation of enzyme. Any further increase will not cause any change. At this stage, velocity or the rate of reaction is maximum, called as maximum velocity or Vmax. So here one thing you just understand that whenever we increase the substrate concentration, so here x-axis is low substrate concentration on the y-axis is denoting the rate of reaction. So you can see here, whenever you are increasing the substrate concentration, rate of reaction is also increasing. So this occurs up to what point? Up to this point. So this is known as the Vmax. So Vmax means, so what is happening here? After this point, even though you increase the rate of reaction, substrate concentration, rate of reaction increase kaadu. It remains constant. So this point is the Vmax we call. That is the velocity, maximum velocity of that reaction. So what is happening here? Whenever we increase the substrate concentration, whatever active sites are available in the enzyme, all those active sites are filled with the substrates. So whenever that point is reached, when all the active sites are filled with the substrate molecule, that is the enzyme substrate complex is formed. So that is the rate, maximum rate of reaction, velocity of reaction you can say. So that is denoted by Vmax. Okay, so this is very important, Vmax. Then the presence, another is the presence of inhibitors and activators. So inhibitors, as we discussed earlier, are those chemical compounds which decrease or stop the chemical activity of enzymes. So we discussed earlier three types, that is the competitive inhibitors, non-competitive inhibitors, and feedback inhibitors. So here you see here inhibitor. So here what is happening here is, this is the enzyme. This is the active site here, and this one is the allosteric site, and this is the inhibitor. So whenever an inhibitor combines at a allosteric site, the active site, what happens here is active site gets changed. So you can observe here. So ఇక్కడ అది change అయిపోయింది. So the substrate molecule cannot come and bind with the enzyme, and substrate enzyme substrate complex is not formed. And whenever this inhibitor is binding here, ఇక్కడ bind అయితా ఉంది కాబట్టి allosteric stage లో, there is a change in the active site of the enzyme whereby the substrate cannot bind with the enzyme, and so this active site is altered and the product is not formed. So that is how the inhibitors affect. So inhibitors, they may be an competitive type, that is the first one. Then second one is the non-competitive inhibitor. Third one is the feedback inhibitor. So that is how inhibitors have effect on the enzyme. Whereas activators are there, where this is the enzyme and this is the activator, and this is the altered active site. So what happens here is, whatever an inhibitor does here, that is it combines at a allosteric site. So same thing, there are few activators also. What does this activator do? whenever this activator combines to the enzyme at a allosteric site, so that creates a positive change in the enzyme. So what is that positive change means? It alters the active site, that is similar to the substrate. So that is how whenever an activator combines to the enzyme at an allosteric site, it creates a positive impact on the enzyme at a and that is how it alters the active site suitable for the substrate, and the active site is altered and the substrate enzyme substrate complex is formed, and we can get the product. Okay, so this is the impact of effect of inhibitors and activators on an enzyme. So what is happening here? In one way, inhibitor completely blocks the enzyme. It does not allow the enzyme substrate complex to form, and products are not formed. And whereas the activators, they act opposite. It activates the enzyme by attaching it at a allosteric site. Enzyme substrate complex forms due to the change in the active site, and we are getting the product. So this is the effect of inhibitor activator. Okay. So to sum up, a number of factors affect the activity of enzyme in an enzyme catalyzed reaction. So the main factors are temperature, pH, substrate concentration, presence of inhibitors as well as activators also. Okay. So temperature, what it does it do? Enzymes are thermolabile, means sensitive to temperature. They are sensitive to temperature variations. The range of optimum temperature is between 25 to 40 degrees centigrade. Increase in temperature beyond the optimum temperature or a high temperature causes change in the shape of active site, and ultimately loss of enzyme activities occurred, which is known as the denaturation of enzyme. Then the second is pH. Each enzyme has an optimum pH ranging between 6 to 7, 7.5. So beyond this pH range, the enzyme become inactive. That is denaturation of enzyme occurs. Then the enzymes are denatured at extreme soft pH. That is, even though if the optimum pH is 6 to 7, even if it falls below that range or it is above that range, then the enzymes are denatured and they become inactive. Then the another one is substrate concentration. At constant enzyme concentration, increase in substrate concentration increase the rate of reaction. This can happen till what? Up to all active sites are occupied. Then the substrate is known as saturation of enzyme. Any further increase will not increase in any change. So that point is known as what? V max. Okay, it is known as V max. That is velocity at maximum. And last one is the presence of that is the inhibitors and the activators. So inhibitors, they prohibit the enzyme activity, whereas activators, they promote the enzyme activity. So with this, we end the enzymes chapter, and I stop here. So we'll start with another new chapter in our next class. So I want you all to quickly just switch on your videos. We are left with another 2 3 minutes, 2 minutes. Just switch on your videos so that I can take a screenshot. Tarak, Bharati, Dilip, Mary, Nagaveni, Sham Kumar. The videos on cheyandi. And if if you want to ask anything, you can ask. If you have any doubts, you can ask now. Unmute yourself and ask Switch on your videos so that I can take a screenshot. The video is on చేయండి for 1 second. తారక్, మేరీ, భారతి, నాగవేణి ఒకసారి మీ వీడియోస్ ఆన్ చేయండి. మేరీ ఫ్లోరా, నాగవేణి, శ్రీభారతి and Tarak. Mary Flora and Nagaveni, switch on your videos for 1 second. I want to take attendance. So I'm once again telling that you sit in a place where the signal strength is very good and you can note down the points or you can take the screenshots. Afterwards, I will share the PPT as well as I will share the video link also. You can see the video, whatever we discussed. If you have any doubts, you can post me in the WhatsApp group so that I can clarify it in the next class. Okay, switch off cheskondi, Dilip and Sham. And stay safe at home. Don't move outside unnecessarily. Wear mask. Okay. Thank you all for joining. So we'll meet in our next class today and here. Anybody wants to ask anything? Nothing to ask, then